We'll go ahead and get started. I'd like to welcome everyone to the Center for Engineering Mechanobiology Future Leaders seminar series. This is a seminar series that aims to put a spotlight on early stage and up and coming leaders in the field of mechanobiology. We have a lot of strong PhD and postdocs in our center, and so we want to highlight their work. It's my great pleasure today to introduce our speaker, Richard Vincent. Rich attended New Jersey Institute of Technology and pursued a bachelor's degree in biomedical engineering with a focus on biomaterials, doing some really cool undergraduate studies looking at various polysaccharides, mimics for cartilage tissue repair. He continued his education pursuing a PhD in biomedical engineering, and has co-authored several recent publications looking at artificial scaffolds that mimic plant extracellular environment, and looking at the effects of various types of polysaccharides on chondrogenesis. And so without any further ado, again, I would like to kind of turn over the floor to Richard. Before I do that, if you want to learn more about Rich and other projects, he will be featured on our new CMB podcast. There's a CMB podcast, so super excited about that. With some other CMB students, Ryan from Kokut from WashU. It will be released on Monday. So Richard, the floor is yours.
Thanks so much for the introduction. I'm just going to share my PowerPoint presentation. Let's see if you guys are able to see it.
Not yet.
All right, so are you guys able to see the presentation?
It's not in full presentation mode yet. There you go.
That sounds good. Thank you. Thank you for that great introduction, Dr. Faustin. I'm just going to give you guys a quick talk into some of the work that I've been doing in the lab. This talk is entitled Glycosaminoglycan Mimetic Structural Variation Impacts Stem Cell Chondrogenesis. And this is going to be work that is for this CEMB Future Leaders talk. So in this talk, I'm going to briefly give a quick introduction into some of the background into the work. I'm going to go over sort of two parts of my work. First thing that I'll be discussing is sort of the synthesis, characterization, some of the chondrogenic pellet culture studies that we've run in the past. The second part of the work is going to focus on more of sort of the culture of stem cells on the fibrous matrices that we fabricate, and then I'm going to give you a quick overview of the summary and the future studies. So the ailment that our lab aims to address is osteoarthritis. And osteoarthritis is a condition that affects roughly 41 million Americans. We focus primarily on osteoarthritis in the human knee, which accounts for about 90% of the disease's total burden. Osteoarthritis is a degenerative disease of articular cartilage, which is found in joints. And in the case of the human knee, up to the upper right-hand side, articular cartilage lines the distal femur, the ruptural surface of the patella, as well as the tibial plateau. And it's a degenerative disease of the cartilage, which leads to the following symptoms, which include limited mobility, pain, and lower joint function. The tissue itself functions to serve as a smooth surface for articulation in joints, whilst also serving to transmit the joint loads to the bone below. And when it's damaged, there is often bone-on-bone contact, leading to those symptoms. The tissue is completely avascular and thus has limited regenerative capabilities. And it's worth noting as well that the cells that actually secrete the extracellular matrix for the articular cartilage is still there. The chondrocytes are still there. However, they are too sparse to trigger in tissue regeneration. And currently there's no technology that exists that fully regenerates functional cartilage tissue. Some of the current therapies that are available for this ailment include, but are not limited to, just a couple that I'm showing here. First, debridement, which is the removal of damaged articular cartilage tissue or smoothing of the surface. It's often used in conjunction with sort of the gold standard treatment, microfracturing, which is the introduction of blunt punctures into the subchondral bone below with the aim of the resulting clot forming a cartilage-like tissue at later time points. Another form of therapy that's often used are the transplantations. The one that I'm showing here to the right is autologous chondrocyte transplantation. This process involves biopsy and external culture of chondrocytes extracted from a non-load-bearing zone within the knee. They're then expanded in ex vivo and then re-implanted into the chondro defect. It's worth noting that for all specific transplantations, those techniques often suffer from poor integration with the surrounding host cartilage tissue. And in general, all of the currently used techniques or therapies suffer from formation of fibrocartilage, which is a mechanically inferior cartilage-like tissue composed primarily of collagen type 1, whereas the native articular cartilage is composed primarily of collagen type 2. So in our lab, we use a sort of tissue engineering approach to this ailment, wherein we use biomaterials either from ceramics, synthetic polymers, or natural polymers. And we treat those biomaterials with regulatory factors, such as growth factors. And in the case of chondrogenesis, we use transforming growth factor beta. But you can also use mechanical stimulus as a regulatory factor as well. And then we seed cells onto those constructs, and that facilitates the formation of tissue. In our case, the constructs that we primarily use, shown to the upper right, are electrospun fibrous scaffolds. And we use fibrous scaffolds, and it's very important that because they mimic the fibrous nature of the articular cartilage extracellular matrix. And the cell source that we generally use are bone-derived mesenchymal stem cells, which have been shown to differentiate into a number of lineages, including chondrocytes. As we try to design biomaterials for this ailment, what we want to do is take into account the extracellular matrix of articular cartilage. It's primarily composed of collagen type 2, water-swollen proteoglycans, and the proteoglycan that I'm showing in this slide, shown in the middle of the slide, is the most abundant proteoglycan in the cartilage extracellular matrix, aggrecan. It's often depicted as this very large hyperbranch macromolecule, and it's composed of glycosaminoglycans. Glycosaminoglycans are very important. They are very long chain, typically sulfated polysaccharides. And in the extracellular matrix, they behave as polyanions. They're very large and immobile, and because they behave as polyanions, as a result of satisfying the Donan equilibrium, they often swell. And that imparts swelling pressure onto the overall extracellular matrix, and thus affords the actual tissue with very robust mechanical properties. So that's the mechanical role of glycosaminoglycans in the extracellular matrix. The biochemical function is also very well known, as they are known to facilitate the active diffusion of growth factors, which ultimately results in a growth factor receptor binding. They're often also known to sequester and store growth factors, which can ultimately affect cellular differentiation and proliferation. It's worth noting that right now, it's not necessarily well known in terms of the, design of the glycosaminoglycans, what structural features are more important, or what structural features typically affect chondrogenesis. So that's one of the things that we want to investigate right now, are what are the impacts on specific structural features found in these GAGs. And some of the structural features that I'm going to highlight right now are some of the ones that we're interested in for this specific talk. And I'm going to start off by describing first the degree of sulfation. So to the right, I'm showing two common glycosaminoglycans found in the extracellular matrix, chondroitin 6 sulfate and below heparin sulfate. And what I hope you can notice is that sulfates as circled are substituted per monomer unit. The degree of sulfation by our definition is how many sulfates are substituted per monomer unit averaged throughout the polysaccharide. And in the case of chondroitin sulfate, there's one sulfate substituted per 2 monomer units, so it has a degree of sulfation of 0.5, 1 per 2. In the case of heparin sulfate, there's one sulfate per 1 monomer unit, so it has a degree of sulfation of 1. The other structural feature that we aim to highlight is the specific stereochemistry of the glycosidic linkage. This is the linkage that binds two monomer units together. And in the case of the chondroitin sulfate, it's primarily beta linked. And by definition, for beta glycosidic linkages, the neighboring monomer unit is bound within the same molecular plane as the previous unit. The other geometry for the glycosidic linkage is the alpha glycosidic linkage bond, which is shown below in the heparin sulfate. As you can see, it alternates between alpha and beta glycosidic linkages. In the case of alpha glycosidic linkages, the neighboring molecule is bonded either above or below the plane of the previous molecule, and that ultimately leads to changes in the shape of the polysaccharide in general, which can lead to changes in the interaction with growth factors. And the last structural feature that I aim to highlight is the substitution of the other chemical groups to the monomer ring. In this case, I'm circling the acid group and the N-acetyl group. And for the purposes of this talk, we're only going to be talking about the N-acetyl substitution. So taken together, those are some of the structural features that I aim to highlight in this talk as it pertains to the design of the specific glycosaminoglycans. So some of the glycosaminoglycans that are present in the extracellular matrix during early chondrogenesis are chondroitin 6 sulfate and heparin sulfate. We were initially interested in using these, however, they have their drawbacks. They're often isolated from animal sources, and the complex chemistry that's involved in those processes can lead to the presence of impurities in the isolated compounds, potential heterogeneity in GAG content, meaning there can be both chondroitin 6 sulfate and traces of a different GAG in the isolated product. And also this can lead to variations in the degree of sulfation. And we can address some of these potential drawbacks by synthesizing our own glycosaminoglycan mimetics. And the chemical process would allow us to control, or at least have better control over the degree of sulfation. And we can have a little bit of enhanced control over the variation in the degree of sulfation and also the purity. So what we aim to do is synthesize glycosaminoglycan mimetics and use those synthesized mimetics to generate constructs for tissue engineering. So some of the candidate materials that we chose for generating this library of mimetics have to highlight also some of the structural features found in the native GAGs. And again, that's simply the beta glycosidic linkage, or they have to highlight the the presence of the alpha glycosidic linkages. And also one of them has to highlight the presence of the N-acetylglucosamine. And the first candidate material that we chose was cellulose. It's a very abundant polysaccharide. And it's primarily the beta-linked glucose polymer. It's a primarily beta-linked glucose polymer, and we can sulfate that to form cellulose sulfate, the first mimic. The antithesis to that is GU starch, which again is a glucose-based polymer, but it's primarily alpha-linked, and we can sulfate that to form starch sulfates as mimetics. And the last candidate material is chitin. And chitin is often isolated from shellfish, and it's literally the polymer of N-acetylglucosamine. So that is one of the mimetics that we will use to highlight the presence of the N-acetyl group. And again, we can sulfate these to form chitin sulfates. And we generate this library to answer the proposed question, what specific set of structural features is more optimal for chondrogenesis? That led to the hypothesis that structural variations in GAG-mimetics will influence MSC chondrogenic differentiation and cartilage matrix formation. So what I'm going to show here is sort of the general synthetic mechanism for the actual chemistry used to sulfate these polysaccharides. And there's plenty of potential sulfating agents that can be used, but what I'm showing here primarily is the pyridine-based sulfating agent, and the specific reaction mechanism that I'm showing is of the kinetically controlled Markovnikov-like product, which forms at carbon 6. So again, there's the reaction with the hydroxyl of the sulfating agent, in this case is pyridine sulfur trioxide. Following that, what generally happens is the formation of the pyridinium salt of the polysaccharide, which we then saponify off, forming the sodium salts of the sulfate esters. And then we release the pyridine. We're able to afterwards use a series of purifications in order to isolate a purified product. And again, we do that with all three of the starting materials, and we synthesize them in 2 overall degrees of sulfation. So showing some of the characterization data, what I'm showing on the left is the Fourier infrared spectroscopy. And what I hope you could appreciate about these data, again, is that compared to the control, which is the non-sulfated starting material shown at the bottom of all of the spectra, is that as you increase the degree of sulfation, again, we have about 0.5 sulfates per monomer in some of the lower degrees of sulfation variants, and we raise it as high as about 1 to 1.5 in some of the higher degree of sulfation variants. What I hope you could appreciate is that as we increase the overall degree of sulfation, the peak intensity associated with the sulfate peak at about 1220 increases in intensity as well. And we confirmed this finding by running complementary Raman spectroscopy. And the Raman spectroscopy shows that at a Raman shift of about 1050, which is again associated with the sulfate group, all of the sulfated variants show presence of this group, except again the control used for this, which is cellulose. After we ran that, we wanted to check that our purification processes worked well as well. So what I'm showing are two representative proton NMR spectra of the impure, either pyridine-based chemical reactions for starch sulfates and the other dimethylformamide-based sulfating agent base chemistry that we used. And what I hope you can appreciate is that after the purification processes, those peaks associated with the impurities are greatly diminished, and we were able to isolate compounds at a purity of less than 1 mole% impurities. And down below, our elemental analysis also shows that for the starch and the cellulose-based mimetics, again, we have 4 total, because we did it at 2 degrees of sulfation. We're able to get a good separation of the degree of sulfation, increasing this degree of sulfation from about 0.5 to about 1 to 1.5 for those. However, for the chitin-based variants, it was harder to get a lower degree of sulfation, as both of them are around 0.9. So this is some of the this is some further characterization, and I'd like to thank Dr. Falston. This was done in collaboration with them as well. And we ran some carbon NMR of the sulfated variants to analyze exactly where these sulfates were formed. And what we can appreciate about these spectra, and I'm going to go very briefly over this, is that we're able to achieve sulfation primarily at carbon-6, but we were also able to achieve sulfation in some of these compounds as well at carbon-2. And we're still analyzing this data to see if we can further investigate the quantity of the sulfates at specific carbon nuclei. So afterwards, after we did the chemical characterization of those sulfated compounds, what we aim to do is to look at the cytocompatibility of these compounds by looking at just two-dimensional culture on TCP, these compounds in growth media with mesenchymal stem cells. So what we did is we seeded a low number of mesenchymal stem cells onto those plates, and then we supplemented them with media containing increasing concentrations of the synthesized glycosaminoglycan mimetics. And our lowest concentration was down to 0.0001%, and that's weight per volume of the glycosaminoglycan mimetic in growth media, all the way up to 0.1%, which is quite high. When we look at cell growth, I hope you could appreciate is that for the cellulose and for the starch-based GAG mimetics, at very low concentrations, it significantly increases the cell number compared to the growth media control. And for those specific compounds, when you increase the concentration in the media, the cell number is decreased, and this is potentially due as well to limited cell binding. So higher concentrations of the GAG mimetic should be, we have to be careful with the amount that we use in the media. But what's interesting is when we looked at the chitin-based materials, this trend is reversed. Higher concentrations of the chitin sulfate in the media, we actually see significantly higher cell number compared to the control as well. And for clarification, the images that I'm showing are for all of the cells treated with the higher degree of sulfation values. So after we ran that study, what we wanted to do was quickly run a chondrogenic study using cell aggregates, also called micromass or pellet cell cultures. In order to do that, what we did was we first expanded mesenchymal stem cells and seeded them at about 200,000 cells to form the pellets, and then afterwards we centrifuged that down, forming the pellets. And these pellet cultures were supplemented with media containing 0.01% of each GAG-mimetic, so 6 groups total. And we allowed that culture to go up to 28 days. And then afterwards, we isolated the pellets and we analyzed the matrix deposition by ELISA. And again, the components that we were looking for in matrix deposition is collagen type 2, collagen type 1, and we were also looking at sulfated GAG production, and we analyzed that by DMMB. So what we analyzed at day 28 is when we looked at the cell number, we observed that for the cellulose and starch-based GAG-mimetics at higher degrees of sulfation, we saw that there was significantly lower cell number compared to the control. But when we looked at the dimethylmethylene blue assay and analyzed the overall sulfated GAG production and normalized the amount of sulfated GAGs to cell number, we saw that there was significantly more sulfated GAGs produced for those groups relative, again, to the induction media absent any of the additional GAG-mimetic variants. So afterwards, we looked at, again, the matrix production. In this case, we're looking at ELISA for collagen type 1 and collagen type 2. And what I hope you can appreciate is down to the lower left, when we analyze the production of collagen type 2, for the starch and the cellulose-based variants that were supplemented in the media, there was significantly higher production of collagen type 2 compared to the growth media control, as well as the chitin sulfates. And when we looked at collagen type 1 production for partially sulfated cellulose, there was significantly higher production of collagen type 1 for those groups. And then when we, what we do is we look at the ratio between collagen type 2 and collagen type 1, which is again associated with a more homogeneous articular cartilage-like matrix. We saw that the group that had the highest collagen type 2 to 1 ratio was actually the starch sulfate group at a degree of sulfation of 1.5. We then analyzed some pellet histology, and again, across the board in these, we saw strong proteoglycan staining using Scaffold for these pellets treated with the GAG-mimetic variants. We also saw very, I would say, not intense staining of collagen type 1 within these pellets as well, but we saw very strong staining for collagen type 2 when we treated these pellets with the glycosaminoglycan mimetic variants. Afterwards, we took the purified variants after we analyzed the standard model for chondrogenesis to pellet cultures, and we fabricated scaffolds, and we blended the glycosaminoglycan mimetic variants with bovine gelatin in the ratio of 10% of the GAG mimetic per scaffold, and the rest was composed of gelatin. We then electrospun those scaffolds, which is the application of charge to the polymers to a polymer solution. The fibers then erupt from the needle charged with that voltage, and the solution then undergoes high elongation of flow and then rapidly vitrifies, forming micron scale fibers. And the resultant scaffold we then cross-link using EDC and HS chemistry. What we were able to do is we were able to produce scaffolds with relatively with similar fibrous architecture. On average, the fiber diameter for some of these scaffolds, were around 2 microns, and the interfiber spacing on average for the fiber scaffolds was about 8 microns. We then looked into the mechanical properties of these electrospun scaffolds as bulk materials using instron testing. And what we observed was that there was a significant increase in the Young's modulus in the scaffolds containing either both of the cellulose-based or both of the starch-based glycosaminoglycan mimetic variants. And again, it's worth noting that the greatest Young's modulus was for that of the starch sulfate containing scaffolds at a degree of sulfation of 1.5. And this is true for the Young's modulus as well as the ultimate stress. So after fabricating those fibrous matrices, we wanted to quickly analyze whether or not these matrices were going to be hydrolytically stable and retain the GAG content that we introduced when we blended them and electrospun them. So in order to do that, we quickly incubated these scaffolds with PBS and continued to change the media every 2 weeks, simulating media changes for cell culture. And we did that up to 90 days in culture. And what I hope you could appreciate is that above, when looking at the scanning electron microscopy, is that some of the fibrous architecture is still retained after 90 days in culture. And then when we look below, we also looked at the glycosaminoglycan content that was released from the scaffold. And in order to get those samples at every media change, when we changed the PBS, we isolated that media and then we just checked it via DMMB. And what I hope you can appreciate is that for some of these scaffolds, there is a very gradual release of the GAG-mimetics within the scaffolds, and only about 50% maximum of the GAG content from the scaffolds is released over a 90-day period, which is very favorable. And typically, it's the release tapers off after, I guess, day 40, right around 1,000 hours. But 3 of the groups were able to retain significantly more of the GAG-mimetic over that 90-day period, which is again the cellulose-based mimetic, and again the starch sulfate at a degree of sulfation of 1.5. So afterwards, what we wanted to do was analyze the ability of these scaffolds to sequester growth factors, which is important for chondrogenesis. And in the case of chondrogenesis, the growth factor that's important for us and that we were using in our induction media is transforming growth factor, primarily isoform beta-3. In order to do this, what we did is we subjected each one of the scaffolds, and we added in 10 nanograms of TGF-beta-3 in the media to the scaffolds, and we isolated the media at each time point to analyze how to indirectly measure the amount of TGF-beta loaded onto the scaffolds per each time point. And what I'm showing are the time points to 24 hours. So what we were able to then do with those data points is fit it to the one-phase exponential association equation, and that allowed us to back calculate a association rate constant. And what we see when we looked at the data is that the association rate constant for TGF-beta was greatest for scaffolds containing 10% of starch sulfate 1.5 or the cellulose-based variant at a higher degree of sulfation. We then took those scaffolds that were loaded with TGF-beta and subjected them to media changes over a 28-day period. And what this allowed us to do was indirectly measure the release of the growth factor from the scaffolds over that 28-day period. And we fitted the data points in this case for dissociation to the one-phase exponential dissociation equation, which allowed us to then back calculate the dissociation rate constant for these groups. And what we were able to observe was that the lowest dissociation rate constant was for that of the partially sulfated cellulose at a lower degree of sulfation based scaffolds. But also, again, we saw that starch sulfate once again had the lowest dissociation rate constant compared to the remainder of the groups. What all of this combined allowed us to do was to calculate the overall affinity of the scaffolds of those constructs to TGF-beta-3. And what we found that was interesting was both partially sulfated cellulose containing scaffolds had relatively high affinity for TGF-beta-3. But the highest overall affinity for TGF-beta-3 was for the constructs containing starch sulfate, a degree of sulfation of 1.5. What we also found that was very interesting was that both chitin sulfate containing scaffolds had a lower affinity for TGF-beta, even compared to the gelatin control that had no glycosaminoglycan mimetic variant within it. So what that led us to suggest was that the N-acetyl the N-acetyl group itself is potentially poisoning the affinity for TGF-beta-3. Again, we see similar data when we looked into our chondrogenesis studies. So just some discussion points for this first part of this talk. So for the chemistry, the sulfation that we, by the NMR, we were able to observe that sulfation was primarily occurring on carbon-6, but we also saw primarily occurring on both carbon-6 and carbon-2. And that specific, the quantity of the substitution between those two may change based on a couple of factors. First, due to changes in the glycosidic linkage geometry, but it also can change based on the presence of branching, primarily for the starch groups, which can ultimately lead to changes in the overall relative abundance of C6-hydroxyls for that reaction to occur. The other thing that we noticed was that GAG-mimetics were shown to be cytocompatible, but at higher concentrations, the GAG-mimetics may negatively affect cell growth and probably also have a negative effect on the cell adhesion. We are also able to show that a higher degree of sulfation mimetic-containing scaffolds often had a higher association rate constant. And the two scaffolds that had the highest affinity overall were for partially sulfated cellulose and partially sulfated starch. And again, for the starch, it was at a higher degree of sulfation. And another thing that we were able to see was that chitin sulfate containing scaffolds have low affinity, and it's possibly due to the presence of that N-acetyl group. So that ends the first talk. And for this second talk, I'm going to be speaking more along the lines of the preparation for the static culture in preparation for the dynamic culture. And the purpose of the static culture was to essentially reduce the group size to show which specific GAG-mimetic containing construct was optimal. And then we used the optimal group for the bioreactor studies. So the first thing the things that we do in order to run these studies is we punch the scaffolds, the electrospun crossing scaffolds into discs. We then seed mesenchymal stem cells onto those scaffolds, and then we culture them, in this case, in induction media for up to 28 days in static culture. And then we analyze, again, the presence of the typical markers for chondrogenesis, which include, again, collagen type 2, collagen type 1 ELISA, imaging for aggrecan, and we also look at GAG production by DMMP. And we also ran gene expression studies. So the first thing we wanted to do was screen down the groups to find which one was optimal for our future evaluation. And in this case, we seeded 300,000 cells onto those scaffolds and cultured them out to 28 days and evaluated the cell number and, again, the matrix production. And in the case of the cell number shown in the middle, what we can appreciate is that for both cellulose-containing scaffolds at both degrees of sulfation and for the starch-containing scaffolds at a higher degree of sulfation, there's significantly lower cell numbers, again, compared to the gelatin alone control. And then when we looked at the matrix production, we saw that, in general, increasing the degree of sulfation resulted in enhanced collagen type 2 production. We also looked at collagen type 1 production, and we saw that for the starch sulfate group at a higher degree of sulfation, there was a significantly lower production of collagen type 1 compared to the other GAG-containing groups. And then lastly, when we ran the ratio, we ultimately determined that the two optimal constructs from these groups were partially sulfated cellulose and partially sulfated starch at higher degrees of sulfation, because they were able to facilitate the higher collagen type 2:1 ratio for those cells. It's worth noting as well, we also ran linear regression analysis, and the goal of that was and what we observed was that there was a significant correlation between the collagen type 2 to collagen type 1 ratio for the cells treated with those scaffolds and increasing the degree of sulfation in the GAG-mimetics used within those scaffolds. So we found that to be pretty interesting. Next thing that we did was we ran some confocal images, and we immunostained all of these the cells at day 28. In these images, shown in red is the actin staining, blue we're showing nucleus, and in green we're showing the protein of interest, which is either collagen type 2 or collagen type 1 or the proteoglycan aggrecan. And from these images, what we were able to see is, again, strong staining for collagen type 2 across the board in these confocal images. And we also saw positive staining for collagen type 1 and aggrecan, and these are all at day 28 for these compounds. So after running that initial study and determining that the partially sulfated cellulose and starch variants are going to be resulted in the highest collagen type 2:1 ratio for that initial study, what we then wanted to do is compare these synthetic glycosaminoglycan mimetics to the native GAG. And although chondroitin 6 sulfate and heparan sulfate are two of the GAGs found in the extracellular matrix, we chose heparin to do the comparison in this case as our native GAG because of a couple of factors. So first, it has a similar degree of sulfation to the mimetics that we're using already, but it also allows us to run a direct comparison between the presence of the specific molecular features, in this case, the glycosidic linkage of the GAGs in those constructs. So again, to reiterate, partially sulfated cellulose highlights the presence of purely beta glycosidic linkages. Sulfated starch highlights the presence of purely alpha glycosidic linkages. And interestingly, heparin allowed us to compare either highly or purely beta, purely alpha, or alternating alpha and beta. So that's why we chose that specific compound as a good control for these next experiments. So again, the first thing we did was we cultured these scaffolds. In this case, we cultured 50,000 cells per scaffold, and we analyzed them at the following time points, days 1 all the way up to day 28. And when we analyzed the cell number, what we observed was that the cell number for the synthetic partially sulfated scaffold containing partially sulfated cellulose or starch containing scaffolds showed significantly lower cell number compared to the gelatin and the heparin controls. But when we analyzed the matrix production for those groups, what we saw again was that there was a significantly higher production of collagen type 2 in the starch sulfate 1.5 groups, and a lower overall production of collagen type 1 in both of the GAG-mimetic containing groups. When the cells were treated, with the synthetic GAG-mimetic containing scaffolds. And then when we looked at the ratio of collagen type 2 to type 1, we saw that both partially sulfated cellulose and partially sulfated starch containing scaffolds showed a significantly greater collagen type 2-1 ratio than gelatin, and especially the native GAG heparin. So this is just some quick imaging. Again, the stains are the same. Red shows F-actin, blue shows nucleus, and green is showing the protein of interest. And what we wanted to, what I hope you can appreciate from these images is that as early as day 21, we were able to show positive stains for collagen type 2, aggrecan, and we also saw positive stains for collagen type 1 for all of the groups, but especially in the partially sulfated cellulose and partially sulfated starch containing groups. So briefly going over the gene data, what I'm going to draw your attention to as I go over this gene data is primarily the two synthetic GAG-containing groups, partially sulfated cellulose and partially sulfated starch at a higher degree of sulfation. And the goal of analyzing this data was to show which one fosters a more homogeneous chondrogenic gene response compared to each other, as we're going to choose which one is the more optimal group to then use in the bioreactor studies. So in the case of the expression of collagen type 1, again, it was very highly, collagen type 1 was very highly expressed for the partially sulfated cellulose containing scaffolds, whereas again, there was significantly lower expression of collagen type 1 in the starch containing scaffolds. Similarly, there was a high expression of collagen type 2 in the partially sulfated cellulose and partially sulfated starch containing scaffolds. However, there was significantly lower expression of COL2 at day 28 for the starch containing group. But importantly, when we looked at the transcription factor SOX9, we saw that at earlier time points, there is a significantly greater expression of SOX9 when the cells were treated with the starch sulfate containing scaffolds. And lastly, for CHAD, again, there was a significantly high expression of CHAD for both groups. But again, there was significantly lower expression of CHAD at later time points in starch sulfate 1.5 containing groups compared to partially sulfated cellulose containing scaffolds. So importantly, we wanted to look at the MMPs, and we saw that in both cases there was high expression of the MMPs, specifically MMPs 2 and 13 in both scaffolds at both days 21 and 28. But the important thing that we wanted to check was, again, the expression of hypertrophic markers, and in this case, we were looking at VEGF and COL10. And at later time points for both of those genes, starch sulfate containing scaffolds resulted in a lower expression of those genes at later time points. And this is in comparison to the partially sulfated cellulose as well as the gelatin and heparin. So with that data, we've chose to continue on to dynamic culture using the starch sulfate 1.5 containing scaffolds. So in order to perform these dynamic cultures, what we use is we use a bioreactor, but in order to, we use a Cardigan X9 bioreactor. And to generate scaffolds for that, we generally electrospin the scaffolds quite thick. And if we can't get them thick enough, we stack them to a thickness of roughly 3 to 5 millimeters. After we achieve those scaffolds, what we then do is we seed about 1 million cells per scaffold, and we allow the cells to adhere to those scaffolds for about an hour or two. Afterwards, we place those scaffolds into the bioreactor system and only subject them to perfusion. And what this allows for sort of pre-culture for the cells to deposit matrix, and allows the overall construct to be robust enough to withstand the dynamic compressions without falling apart. So we subject them to perfusion alone for 14 days, but then afterwards, for the next 2 weeks, we subject them to the scaffolds to dynamic compressions, and the parameters that we use are dynamic compressions at 1 hertz, about 10% strain for 3 hours daily, 5 days a week, for the last 2 weeks of culture. So what I'm showing now are the groups that were being used. Again, we used perfusion as a control here. So we had both starch sulfate containing scaffolds at a degree of sulfation of 1.5. And the control for those were gelatin scaffolds. And we treated both of them with either perfusion alone as a control or dynamic compressions. And when we analyzed cell number, what we saw is that both starch containing groups, again, treated in both ways, either perfusion alone, which we call PERF, or dynamic compressions, DYNE. What we saw was that at later time points, again, the cell numbers within the starch containing groups, and when treated at both conditions, was significantly lower than that of the gelatin containing scaffolds. But again, when we looked at the sulfated GAG production, what we saw was, again, the opposite, that when we normalized the cell number, there was a significant increase in sulfated GAG production for the starch-containing scaffolds, in particular when treated with dynamic compressions, but in both perfusion alone and under dynamic compressions, you get significantly more GAG production per cell. Then we looked at the compressive modulus of the materials at day 28. There was no significant difference between the groups, and also what I want to make clear is that these isolated materials do not show compressive modulus on par with the native tissue. It's actually quite less stiff than the native articular cartilage. So lastly, again, with these bioreactor samples, we wanted to evaluate the specific matrix composition for these scaffolds. So again, we ran ELISA and analyzed the collagen type 1 and type 2 production, then took the ratio of those, and what we could appreciate from this is it's quite clear that the starch sulfate containing scaffolds at a higher degree of sulfation, subject to dynamic compressions, showed significantly higher collagen type 2:1 ratio above the starch containing scaffolds treated with perfusion alone, and both of the gelatin treated groups. Both of the gelatin groups treated with either perfusion or dynamic compressions. And you can see the effect of the dynamic compressions as early as about day 21 with the starch sulfate containing scaffolds. And when you look on the right and look at the imaging, we saw very, very strong staining for collagen type 2 within those SS 1.5 DYNE treated scaffolds. But we also were able to show positive staining for collagen type 2, a little bit less to a lesser degree positive staining for collagen type 1 and aggrecan for all groups treated in these bioreactor studies. Lastly, again, we looked at the gene expression for these scaffolds, and what I'm going to draw your attention to in these, just to briefly go over it, is essentially the effect of dynamic compressions on both the gelatin and the starch-containing scaffolds. So looking at the aggrecan in the upper left-hand side, the gene expression for aggrecan, as a result of the dynamic compressions at day 21 for the starch sulfate DYNE groups, we saw that there was a significant increase, again, relative to the gelatin in perfusion and the gelatin in under dynamic compressions for the aggrecan expression. Similarly, in the collagen type 2 expression, we see at day 21, again, very high expression of collagen type of COL2 at days 21 and 28 compared to gelatin in perfusion, starch in perfusion, and the gelatin under dynamic compressions. For collagen type 1, for the gene expression for collagen type 1, what I hope you could appreciate is that for the starch-treated groups, especially for starch SS-dine, starch sulfate, subject to dynamic compressions, after the onset of dynamic compression, the collagen type 1 expression decreases at day 21 relative to day 14. Similarly, when we look at Chad signaling, the starch sulfate groups both have significantly higher expression of Chad at days 21, at day 21 for perfusion and day 28 for dynamic compression compared to gelatin and perfusion. But there was also high expression of Chad for the gelatin groups under dynamic compressions. And lastly, SOX9 expression was shown to be high at the later time points for the starch sulfate groups, both in perfusion and under dynamic compressions relative to gelatin scaffolds treated in perfusion alone. Lastly, when we looked at the hypertrophic gene expression for VEGF and collagen type 10, what we observed was that VEGF expression was still was relatively low for all groups except starch sulfate groups treated in perfusion at about day 21. When we looked at COL10 expression, we see that there is enhanced COL10 expression in the starch sulfate containing groups under dynamic compressions as early as day 21, but it was also observed at day 28 in perfusion alone studies where the gelatin group showed reduced expression comparatively. Lastly, when we looked at MMP13 expression, the gelatin-containing scaffolds treated with perfusion alone showed high expression of MMP13 at early time points, but at later time points, the starch sulfate groups under dynamic compression showed higher expression of the MMP13 at days 21 and 28. And again, that's relative to gelatin and to starch sulfate and perfusion. So some discussion points for this portion of the talk. So firstly, the partially sulfated cellulose and partially sulfated starch containing scaffolds enhance chondrogenesis, and we showed that as determined by the enhanced collagen type 2:1 ratio. And it correlated with a higher degree of sulfation of the GAG-mimetics within the electroresponse scaffolds. At higher degrees of sulfation also, starch sulfated and partially sulfated cellulose promoted chondrogenesis, but the cell numbers were shown to be slightly lower than the gelatin alone scaffolds, as well as the scaffolds containing the native GAG heparin. When we utilized starch sulfate at a higher degree of sulfation, we showed lower expression initially of the hypertrophic markers compared to cellulose and compared to heparin, which we utilized to choose that group as the optimal construct for bioreactor studies. Within a dynamic culture, the starch sulfate significantly increased the collagen type 2:1 ratio compared to gelatin and compared to starch sulfate itself when it was subject to dynamic compressions. And this can potentially be due to the mechanical loading, but also the high affinity for the growth factor TGF-beta as we characterized earlier on. So in conclusion, we were able to synthesize a small library of glycosaminoglycan mimetic variants from cellulose, starch, and chitin in 2 overall degrees of sulfation. We were able to observe that a higher degree of sulfation and the alpha glycosidic linkage geometry are structural features that may be optimal for chondrogenesis. But again, what I think is important to show is that either purely alpha or purely beta glycosidic linkage geometry seems to work just fine as well. These studies demonstrate overall the potential use of glycosaminoglycan mimetics at various levels of sulfation for tissue engineering constructs for osteoarthritis. So some of the future studies that I think are important to run, as we weren't able to address everything that we wanted to, was firstly, there was we were only able to achieve 2 overall degrees of sulfation for some of these semi-synthetic glycosaminoglycan mimetics. So one of the things I think is important is to further optimize the sulfation parameters such that we can better isolate an increasing range of degrees of sulfation. And that can be used in order to rectify one of the weaknesses of the study, in that there wasn't enough data points to run essentially a principal component analysis, which would strengthen, I believe, the study quite a bit, and it will give us a little bit more statistical strength when we determine what specific structural features are more optimal for chondrogenesis. Another thing that we aim to sort of rectify is the fact that as you increase the degree of sulfation, as we've shown by GPC, technically you tend to degrade the base polymer and lower the overall molecular weight. So the synthetic scheme could be further optimized to hopefully have a reduced effect on molecular weight and allow, again, one of those studies to be run with the molecular weight hopefully not being a factor that can affect these some of the results that we got. And lastly, what I think was important to do is to electrospin fully sulfated variants of the cellulose and starch groups and evaluate them in vivo and evaluate them in vivo in a cartilage defect model, hopefully in a large enough animal, sort of like a rabbit first, and then we can increase the size from there. But I think testing these in vivo is definitely one of the next steps. So with that, I'd like to thank my lab, who has been extremely helpful in helping me to perform this study. They always give good advice and have been able to deal with some of my shortcomings as well as we went through this process. I also like to thank the collaborators who primarily helped me with the chemistry for some of these. And again, Dr. Falston, I'm going to give you a shout out for helping us with the carbon NMR, which allowed me to complete the process. And again, to CEMB, I'd like to thank CEMB for allowing me to be one of the trainees. It's been a quite unique and very helpful experience overall. And with that, I'd like to end the talk, and I'd like to thank you guys for listening to me. If you guys have any questions, I'd love to hear them.
Thank you, Richard. That was very great. Nice talk. So yeah, we have some time for questions. Feel free to unmute or raise your hand and or type something into the chat. It looks like we have a question from Becky.
Hey, really nice job, Rich. It's sort of nice to see a student progress from the very start to the end like this. Really lovely job. I have a question about when you started doing, in particular, your dynamic cultures, and you noted that there was an increase in collagen deposition. Did you look at all at the collagen organization, like fiber diameter, or any metric like that? Because there's a lot of evidence that different GAGs or different proteoglycans might be important in that, and I'm just wondering what your synthetic ones do.
I see, yeah. So that's a great question. So we didn't necessarily look at the organization. We just quickly did some measurements of the overall deposition of the digested samples by ELISA, but we imaged them as well, just using immunostaining. But we didn't necessarily look at the organization. I think that's a good thing to look into as well. Right. Thank you.
Any more questions? While we wait for another question, I guess I can ask one as well. So you're adding in these GAGs. What do you know about the surface of your fibers, right? Because you have some kind of compositing, or you have some mixture between gelatin and the GAGs, right? And so, what do you know about the surface of your fibers?
Yeah, that's a very good question as well. We did some sort of basic surface chemistry sort of analysis using the FTIR and the Raman, but we also looked at EDXA, and what we saw is that I'm not showing that data there. Is that we saw that it's present, but when we looked at the mapping, it's not. There's a homogeneous, I guess, distribution of it, but there can be areas where the GAGs potentially phase separate from the gelatin that they're in. So you end up getting sort of these areas where they're phase separated GAG-mimetic, but we haven't analyzed that a lot, but we're definitely interested in some more of the sort of presentation of the GAG itself at the surface of the fibers as well. So I think that's something that we should also look into further, but that's We addressed it just barely with some EDXA, but nothing past that.
Okay, cool. I think Charlie has his hand raised.
So I'm curious, maybe in a slightly related question about how the extracellular matrix might be rearranged during the dynamic culture rate, the compression culture, and whether that leads to a phase separation at all. So do you see more phase separation at the end than at the beginning of the culture?
So that's also a good question. We didn't run the EDXA at the after of the constructs treated under the bioreactor. So I think we haven't run that yet, so I can't really comment on it. But again, I think that's something that we should address as well. Thank you.
Any other questions? Well, I like to take another opportunity again, Richard, to kind of really commend you on, all the work that you've done so far. Very impressive, and your presentation, reflected that. So I think I can speak for everybody in the center to say that we're really proud of you, and we're really glad to see you've been able to progress, and excited to see what you do next, actually. So.
Thanks so much. Thanks for everybody. Thanks to everybody in the center as well. It's been a blast.